Now lab animals are no longer necessary for research, and diseases can be studied on lab-grown human tissues. Thanks to Dr. Hao Liu and his team at ETH Zurich, that future is closer than ever. Dr. Liu has pioneered a groundbreaking laser technology that can create tissue structures that mimic the natural architecture of human muscles, tendons, and nerves. So, how does it work? Using a technique called the filamented light or FLIGHT 3D bioprinter, Liu's team harnesses the laser's inhomogeneous or uneven light to create aligned microfilaments. These delicate threads replicate the structure of real human tissues. It's almost like printing muscle fibers in real time. Each filament is only 2 to 20 micrometers thick, comparable to the fibers in our own muscles and tendons. As these filaments form, they create tiny channels that serve as pathways for cells to grow and align, just like in real tissues. By introducing human cells to this gel scaffold, the cells follow the structured channels, growing into fully aligned tissue. These lab-grown muscles and tendons don't just look like the real thing, they function like it too. One of the biggest impacts of this innovation is in drug testing. With these lab-made tissues, scientists can now model human diseases and test treatments without using lab animals. That means faster results and a more ethical, efficient research process. ETH Zurich has already patented this technology, and Dr. Liu's team is working to bring this new bioprinter to market, paving the way for its use in medical research, surgery, and even lab-grown meat production. As Dr. Liu and his team push the boundaries of what's possible, they're not just creating lab-grown muscle, they're building a new future for medical research and beyond. If this breakthrough excites you, don't forget to like and subscribe. Deep Sheet for more insights into the world of tomorrow. Thanks for watching.